Thank you very much. So actually, nociception is just a difficult word to say. That organisms are usually the ability to detect, to sense harmful factors from the environment. And basically, our Blue Waters project is about understanding the molecular mechanism underlying this ability. Now, before I jump into that, I'd like to take a second to introduce the postdoc who's actually interacting on a daily basis with the Blue Water machine, Marina Kasimova. Unfortunately, the slide is a little bit messed up. But the email address was fine. That's the important part. And another second I would like to take just to, let's say, go through a few basics idea about neuronal excitability. So the first important fact is that the membrane on neurons are studded with membrane proteins called ion channels, whose function is to allow permeation of ions through the membrane barrier. And this fact is at the basis of transmission of signals across long distances. And to be brutally brief about that, the fact is that across the membrane of neurons, there is a local charge and concentration imbalance, such that if membranes were made instantaneously permeable, sodium ions would come in the cell, depolarizing the membrane. So let's say counteracting this charge imbalance, while potassium ions would go in the opposite direction and repolarize the membrane. So this is the basic mechanism used by voltage-gated ion channels that are gated or activated by a depolarization, and in such a way as to, let's say, respond to a first stimulus and then depolarize the membrane, repolarize it with potassium channel, and so on and so forth, in such a way as to propagate a specific waveform called the action potential. Now, all this has been studied for 4 decades at least, and you can understand the challenges in the molecular mechanism of so specialized machines that they have to be selective and yet activated at a special, very particular value of the transmembrane potential. Now, what I want to convince you about is that apart from transmitting the signal, there is another class of channels who are specialized instead in generating this signal. So the first perturbation in this transmembrane potential that gets propagated. And these channels are somehow responsible to transduce some stimulus from the outside world into an electrical current. So, for instance, these transient receptor potential channels, I'll call them TRP from now on, so are located in peripheral nerve neurons. They are close to the skin barrier. And what they do is they are activated by diverse stimuli like heat, pH, let's say abundance of protons, reactive chemicals, and so on and so forth, and generating this signal that then, through other structures, get through the brain. So basically, this is the basic mechanism by which we form sensations. An organism is able to somehow figure out what's the outside world. From an evolutionary point of view, I mean, a dendrogram containing a large fraction of the extant ion channels shows that these TRP channels are relatively closely related to the channels I was talking about before, the voltage-gated ion channels. So to the point that they really share the same structural architecture. So basically, during evolution, at some point, voltage-gated ion channels were sort of discovered, and then variations on the team created these TRP channels that differentiate further to detect more and more diverse stimuli. So to the point that So there is a very long list of stimuli that get transduced into currents, and I mentioned temperature and pH. But for instance, ligands like the pungent molecules in mustard oil or wasabi or garlic, or from chili peppers, or the menthol from mint, and so on and so forth. All these chemicals from the external world are detected, as long as others that are instead endogenous, so they are produced inside the body, and there are important ones are some of them synthesized during inflammatory processes. There are lipids, and so on and so forth. Now, the thing that should be mentioned is that all these stimuli get transduced in something that is perceived as a signal of pain. And the important fact about this is that in conditions like chronic pain, so these channels are hypersensitive, and so there is this condition called hyperalgesia, which is, let's say, an excessive response to all of this, which results in a constant and excruciating pain. So that's why this is this class of channels is a very crucial target for pharma industry. So the drug design in this field is actively and currently pursued, but so far no successes. And the problem is that these channels are very, very complicated. And to get you an idea of the challenge, I'm going to focus on just one. So TRPV1, a member, which responds to this ligand, capsaicin, the ingredient of hot chili peppers, but also to high temperature. So it's no chance that I mean, so the brain can tell. We say that this is hot chili pepper because the brain can tell if it is this or that. And as I was saying, this same channel, the single channel, can respond to this, to high temperature, to protons, calcium, and so on and so forth. Very, very complicated. And in addition to that, the presence of one factor alters and lowers the threshold for activation by another factor. So they're entangled, difficult to set apart one from the other. And most importantly, while we do have an idea, a conceptual framework to treat activation by a ligand, so we can say that we have two conformations of the channel. One is conductive, the other is not. And let's say the conductive one offers some favorable interactions for the ligand, and therefore gets selected in presence of the ligand. As far as temperature is concerned, we basically know nothing. So we have no idea about the possible mechanisms. All right. So that's why the goal of the project was to use molecular dynamics to get some insight on that. And as all molecular dynamics projects started with structure. So cryo-electron microscopy structure of this channel. And from these cartoons, I would like you to appreciate the fact that the channel is very big. It extends much more than just the transmembrane part that is this one. And the second part is that it is fourfold symmetric. So these symmetric units here are colored in different colors, but the channel is just a repetition of them. So of course, I mean, these experiments have been done at the temperature of liquid nitrogen, so they cannot say much about temperature activation per se, but the experiment is extremely useful in terms of information, because they were able to capture the channel in three distinct conformations. One is the so-called apo channel, so low temperature, no ligands, the channel is closed. And the second one is the channel in presence of capsaicin, which is not able per se to activate the channel at low temperature, but it is at high temperature. And then the third one is just an open state, so a channel in complex with ligands that open the channel no matter what the temperature is. So at this point, we tried to put together all these things and try to see if molecular dynamic simulation was given any insight about that. And these are the key aspects of the project. So with these points, I'm trying to summarize a little bit what I've just said and give you a little bit the guidance for what follows, which will be mostly results. And as I said, the key challenge is to come up with a molecular mechanism for temperature sensitivity, which is more than a simple cartoon, something that is quantitative enough to be experimentally testable by electrophysiology. And it's important because despite the fact that virtually every organism has to sense temperature to survive, we don't have any viable model at all. So blue waters is kind of crucial for this because the system is big. We need to run atomistically detailed molecular dynamic simulation of systems on 500,000 atoms. And we need to explore several a wide range of parameters in temperature, the presence of ligands, different starting points. So we need both. Run long simulations on large systems, therefore a parallel scalability, which is good, but also a reasonable amount of capacity to have different jobs going on. So what we got so far is a preliminary model that seems viable, and we need to refine, but it's very close to something that can be proposed to the experimentalists. So the Blue Waters team helped a lot, and Victor Anisimov in particular helping us compiling GROMACS in a decent way. And about the broader impacts, so the societal issue of pain is relevant, and the fact that studying this channel can lead us to non-opioid anti-pain drugs. I briefly touched upon this point before. All right, so so the project really started by a relatively long exploratory molecular dynamic simulation of the channel embedded in a lipid bilayer and the water and so on and so forth. And the first thing that we realized is that as the structure was sort of changing during this run, and for a couple of subunits of these symmetrically equivalent subunits, as the structure was drifting away from the initial configuration, it was converging toward the one of the open state, this red line. And this was extremely encouraging because sort of suggestive of the fact as we switch on temperature, the channel is kind of waking up and is activated as it is, it goes toward the open activated state as it is supposed to be. And I mean, looking at this a little bit more carefully by pulling all the a couple of simulations together, all the configurations from different trajectories together with experimental structures and doing a multidimensional scaling projection, it is clear that, you know, as they diverge from the initial structure, some of them goes to the open conformation, some of them stays in the same, other ones as well goes into the apoclosed state. So it seems that we are able to navigate a landscape with several metastable substates. And looking a little bit, looking closely to what happens to the simulation, we see that basically the channel is really opening. So it's the radius of the pore at the constriction side is increasing abruptly, and it is starting to be populated with water. So it's encouragingly similar to an open state with a density of water spanning the entire length of the pore. Therefore, we ask the question, what is the fact that makes the initial structure unstable and makes the channel become open? And we noticed that systematically in all simulations there was an abrupt transition of a specific side chain in the pore, which occurred in the very initial phase of the simulation. So this is an asparagine residue, which by increasing the polarity of the pore, is drawing more water in the pore. So it's reasonable that just as this switch is causing let's say making the channel able to open. So at this point we asked ourselves if a different or a better equilibration of the initial structure could avoid this abrupt transition and allow us to explore better the closed state. So what we did is that we, in the initial phase of the trajectory, we added external potentials to restrain the configuration of these residues to look outside the pore. And then we released the constraints and we noticed that indeed the simulation was stable with these residues fixed or fluctuating in the initial conformation. So with this new protocol, we don't observe anymore the opening of the channel. So the red curve is always above the blue one. And so we started to suspect that we were sampling the closed state of the channel. So indeed, the picture is this one. In one simulation, at some point, there is water starts to populate the pore, and there is a continuous density, while in the other one, a region of the pore here where the gate is stably devoid of water, and it goes on forever. So reinforcing the idea that we have two stable states, one is hydrated, the other is not, and we would like to know now if we do indeed observe a conductive open state and a closed state. And to do so, we need to study ion permeation through this structure. And we used metadynamics to do so, which is a way to sort of introduce an additional time-dependent potential to the equation of motions in such a way as to discourage the system from exploring repeatedly the same region of the conformational space. So this method effectively broadens the scope of the exploration of the conformational state. At the same time, with a little bit of bookkeeping, one can reconstruct the underlying canonical distribution and therefore the so-called free energy profile. So finally, the results are that while the dry state devoid of water, this red line here, has clearly a potential of mean force that doesn't allow permeation of ions. It shoots to infinity here. The other one has barriers that are fairly small and compatible with the experimentally observed conductance. So we are confident now that we did observe indeed a transition between a closed and an open state. Therefore, we went on and asked ourselves if the conformation of this residue is controlling the openness of the channel, what is instead driving the transition between those two conformations? And what we found is that the factor that stabilizes the outward conformation of this asparagine residue is the solvation of a set of pockets, one for each symmetric subunits, that are outside the pore, and they are hydrated at least to the same amount as the pore itself. And this is observed consistently in all the subunits, and you can see from the structure from below that these are rather peripheral pockets that are not in contact with the pore, and yet they are connected to, let's say, the bulk of the solvent on the cytoplasmic side. And So these pockets are not an artifact of the simulation, because they're actually already present in the experimental structure. So one of the structures in particular, the one we were using for the simulation, has a region that is fully connected and can accommodate waters, that is completely devoid of electronic density in the experiment. So we believe that this is not just an artifact of the molecular dynamics simulation. Now, if our let's say model is true, so we should achieve the same effect. Rather than putting restraints on the conformation of the asparagine, we could put restraints or external potentials on the water molecules, so that these pockets are hydrated, and then release the constraints and monitor the stability. And that's exactly what we did here, and we verified that one or the other thing are equivalent. So the simulation are even longer than this, but the message is that, I mean, the two factors are correlated, and that's why we did a much bigger and computationally intensive simulation this time, a two-dimensional free energy surface in which we monitor simultaneously the configuration of this asparagine and something that is related to the hydration of the pocket. And the colors are not, the best ones, but the idea is that there is a metastable state here that is distinct from this basin, and the transition between these two states is what governs opening and closing of the channel. So to summarize, I mean, what we see is that hydration of the pore and of the four pockets is exclusive. So they are alternatively hydrated, and the switch is controlled by the configuration, and in particular by the direction of the dipole moment of this asparagine. So consistent with this view is an evolutionary analysis that shows that really this asparagine is outstanding in terms of conservation within the family of the strips and related subfamilies. And somehow from this we built something that is, just the final one and I'm done, which is finally related to our model of temperature sensitivity. And from this I will use an analogy is what we are doing right now. So we're saying that the hydrophobic hydration of those pockets is the factor controlling the openness of the channel. And in systems of similar size, buckyballs C180, so it has been shown several times, but for the first time in this paper, that not only the hydration shows two states, right? So a dry and a wet state, but the relative equilibrium between the two is sensitive to temperature, and this sensitive also to changes in the interaction between the waters and the wall, like the changes in the conformation of the asparagine. So overall, the idea is that temperature sensitivity of the channel comes from, let's say, a different propensity for being hydrated at different temperature. Okay, and with this I'm done. So thanks, of course, for the computer time to NSF and NCSA, and the project is also supported in part by NIH. And thank you for your attention.